Hello everyone, we'll continue with the topic that is organization and management of pathology laboratory with respect to part 2. General equipments, instruments and reagents. So we'll see how we need to have instruments, equipments and reagents in each area of pathology laboratory. Some general measures are given here, that is we need to have the calibration and standardization of the instrument. So calibration refers to, it is the act of checking or adjusting the accuracy of the instrument. So calibration refers to the act of checking or adjusting the accuracy of the instrument. So whatever instrument we use, we have to adjust it so that it is going to give the accurate values, and standardization of the instrument is done. Proper handling of the instrument, so those who handle the instruments, that is the technologists, they should have the knowledge how to handle and maintain the instruments. Proper maintenance and regular cleaning has to be done. Thorough cleaning of glasswares is very essential, so that if untidy glasswares are present, there is a chance that red cells may undergo hemolysis and there is a chance of getting the false results also. Record of validation has to be maintained. So record of validation with respect to the instruments and equipments as well as reagents, to what extent it will be valid, all those details also should be recorded and it should be maintained. So these are some of the equipments which is being given with respect to phlebotomy area. So equipments also include certain chemicals in this, like alcohol and other equipments like disposable sterile needles and syringes, tourniquets, sterile plain test tubes, bottles with anticoagulants, blood culture tubes, test tube rack, tray, labels, dry cotton swab, vacutainer tubes, etc. So just remember your phlebotomy area so that you will be able to list out the different equipments present in the phlebotomy section. So with respect to vacutainer tubes, we'll just go through what the different colored vacutaners indicate and for what purpose it will be used. Red color, the stopper color, that is the cap of the vacutainer color is indicated here. The additives indicates the respective anticoagulant present and for what purpose these colored containers are used. Red color means no additive, it is used for the collection of serum. Lavender means it is having EDTA, it is used for the whole blood collection. Green means it is having heparin, inhibits thrombin activation. Blue means buffered citrate, it is used for the coagulation studies. Black means buffered sodium citrate, it is used for the estimation of ESR by Westergren's method. Gray means it will be having sodium fluoride, which is a glycolytic inhibitor for glucose, it is used for the glucose determination. Yellow means it is having acid citrate dextrose, which preserves the red cells. So very important to know the color codings as well as the additives and for what purpose it will be used. Next, moving on to the other sections, that is hematology and coagulation section. So here are some list of instruments as well as reagents which will be used in both the sections. Quickly we'll move on. Instruments like microscope, hand tally counter. So what is this hand tally counter? It is nothing but it is a counter where it manually counts the differential leukocyte count. That different types of WBCs can be counted by using this hand tally counter. So in practicals we make use of the 100 squares. Instead of those 100 squares, we can make use of this counter to perform a differential leukocyte count. Centrifuge, water bath, refrigerators, incubators, graduated pipettes, pasture pipettes, blood cell pipettes, test tubes, glass beakers, conical flask, test tube racks, timer, stopwatch, wash bottles, filter papers, chemical balance, glass slides, cover slips, labels, staining racks, spectrophotometer, counting chambers, Westergren's tube and rack, Windroop's tube and rack, microhematocrit tubes, graph paper, disposable lancet, mounting media, etc. So these are some of the instruments which is being listed out under the hematology and coagulation section. So with respect to reagents, just remember the parameters which has been run in a hematology lab, so that with respect to each parameter we can list out the different reagents. like Drapkin solution for hemoglobin estimation, dilating fluids for our cell counts, 1% brilliant crescile blue for the reticulocyte count, Leishman powder, acetone-free methyl alcohol plus the buffer water is used for a smear staining, Pastine Sudan black tea stain, peroxidase reagent 10% potassium ferrocyanate, neutral red absolute alcohol, concentrated HCl 10% formalin, 40% formaldehyde, distilled water. These are all the requirements for a special stain. Thromboplastin reagent, calcium chloride, APTT reagent, thrombin reagent, urea, control and normal plasma, aged serum, adsorbed plasma, factor VIII and IX deficient plasma, D-dimer test kits. All these are required for a coagulation test, that is to perform PT as well as APTT. So in our hematology laboratory, we also have some of the automated instrument. That is, main important one is our automated cell counter. It is mainly used for counting of the cells. Just know the name of the principle, on which principle the cell counter works. It works on the principle, that is the Coulter principle, or it is impedance or conductivity or flow principle. It is also known as light scatter principle. By different principles, the automated cell counter can work. Automated slide making and staining. It is mainly used for making the smears and staining those smears. Automated or semi-automated coagulometers. It is for coagulation study purpose, and this works on the principle of optical density, that is the principle of colorimeter. So OD values will be recorded. So upon clot formation, OD will be taken and the timer will be stopped, so that it is going to read out the prothrombin time as well as activated partial thromboplastin time. Urine and stool sections also have their respective reagents as well as equipments, like plastic disposable containers, reagent strips, pH or litmus paper, urinometer, reagents used for various tests, grams iodine, zinc sulfate, 0.85% saline. These are required for the concentration of the stools. Automated urine analyzer is also present. So automation is possible even with respect to urine analysis. Washing room also have its own equipments in it. Autoclave, hot air oven, distillation plant to obtain the distilled water, ultrasonic cleaner, detergents and other cleaning agents. In office, we need to have these things like telephone, filing cabinets, computer with a printer, stationery items, etc. Other accessories which can be present in our lab, like gauze, solution jars, waste disposable containers, soaps, detergents, paper napkins, towels, incinerators, first aid kit and standby generators. Standardization of equipment and calibration, that is, we are checking for the accuracy of the instrument. So this is being done at the first time of the installation of the instrument. So whether after assembling all the parts of the instrument, whether it is working fine or not, so that it is going to give the required reports. So that is the calibration which has to be done at the first time of the installation of the instrument. Calibrated according to the operational instructions. So how the manufacturer has said to calibrate, accordingly we need to calibrate the instrument. Standardized with standards or reference material or known samples. So calibration, that is, you are checking for the accurate results with respect to some reference materials. Those are called as standards or reference materials, or known samples are nothing but those are the QC samples, quality control samples. Workforce or staffing, that indicates who and all people should be working in the laboratory, like medical officer, where they will be having a degree of MD in pathology. Laboratory technicians, they will be having either degree or diploma in MLT. Technical supervisors, those who supervise the technicians to do their work. Lab assistants, those who help the technicians. Secretary or clerk cum typist to generate the reports or for other computerized works. Personnel management. So how do we manage the persons who is present in the laboratory? by having the regular meetings between the management and staff. So in the meetings, give the chance for the employees to put forward their problems so that the problems can be solved off. Rotation of the staff has to be done with respect to the different sections of the lab, so that this will help them to build their confidence, so that they will be able to perform their work in all the sections of the laboratory. Participation in national or international workshops and CMEs, so it also should be encouraged so that they can update with your knowledge. Proper training schemes also should be available for those who wish to upgrade themselves either for their higher qualification or for their promotion purpose. Standard operating procedures, that is SOPs, it is nothing but it is a laboratory manual. So this laboratory manual should include all the steps which is being followed for any procedure of the parameter done. So all the steps, including the requirements, what procedure is done, how the report is given, all the details should be present in this laboratory manual. It will be useful for the new recruits and trainees, for the new staff as well as the people who are in training. For them it will be very beneficial, and it is prepared by the medical officer or lab in charge. And this SOPs or the laboratory manuals should be reviewed regularly. Either anything has to be added up or it should be deleted. So accordingly the reviewing has to be done. Standard laboratory test results, that means how our reports should be present. So this is the ultimate product which is being generated from the technologist work. So it should be of it also should be in the proper manner. So it is of great value when the patients are referred from one place to another. That means these laboratory test result, that reports may go from one place to another place, so that it should be in a proper format. So this is a format. It may contain these many things in a particular report, like patient's name, address, age, sex, name of the referring doctor, hospital number, lab reference number and date, title of the report like hematology, clinical pathology, whatever it is, investigations requested, patient's test result along with the reference values, signature of the in charge of the laboratories. See that our results are present along with the reference values, and our signature or the in charge signature should be present. Record keeping, it refers to the documentation. Record all the test results performed each day. In one day, whatever test you are going to perform, everything has to be recorded along with its reports. Duplicate copy of dispatch report should be filed. Double copies are there, means see that you won't throw it, instead you file it. Other registers to be maintained like patient's details, date of the sample, collection, date of dispatch of report, test results and payment details.